Hello, today we are going to be discussing DNA replication in a prokaryote organism. Replication takes place in 4 stages: initiation, unwinding, elongation, and termination. In the first stage, initiator proteins bind to the origin of replication. Binding of these proteins will cause a short segment of DNA to unwind, allowing helicase and single-strand binding proteins to attach to the strand. In order for synthesis of new DNA to occur, we need only a single strand, which brings us to the second stage of replication, unwinding. Enzymes and proteins help to unwind DNA, the first being DNA helicase, which breaks the hydrogen bonds that exist between the bases of the 2 DNA strands. Next, single-strand binding proteins tightly attach to the exposed single-stranded DNA, helping to provide stability and prevent the formation of secondary structures such as hairpins, which may interfere with DNA replication. The type 2 topoisomerase gyrase reduces the torsional strain that builds up near the replication fork from unwinding. Once DNA is unwound, the third stage of elongation can begin. This stage also involves a variety of proteins and enzymes. Primase synthesizes short stretches of RNA nucleotides from RNTPs, which provide a 3' OH group to which DNA polymerase can bind. On the leading strand, primers are only needed at the 5' end of the newly synthesized strand. On the lagging strand, DNA replication is discontinuous and a primer is required at the beginning of each Okazaki fragment. DNA polymerase III is a multi-subunit protein which adds nucleotides in the 5' to 3' direction. This protein cannot function unless a primer has been placed by primase. DNA polymerase I follows behind DNA polymerase III and using its exonuclease activity removes the RNA primer placed by primase. Using its 5' to 3' polymerase activity, the RNA nucleotides are replaced with DNA nucleotides. DNA ligase seals the break in the sugar phosphate backbone from where the 3' OH group of the last nucleotide, placed by DNA polymerase I, is not attached to the 5' phosphate group of the first nucleotide, placed by the DNA polymerase III. Telomerase extends the 3' end of a telomere within a chromosome. Termination can occur in some DNA molecules when 2 replication forks meet, or in some there are specific termination sequences which block further replication. If DNA polymerase I lost its exonuclease function in the 5' to 3' direction, the primers placed within the growing DNA strand would not be removed. This would mean that in every replication, both the leading and lagging strands would contain RNA nucleotides at the sites where primers were placed. This concludes my exam number 4 mini video. Thank you for watching.